Hello everyone, and welcome to another video of BioIVT's ADME 101 series. I am Tina Mueller, the Scientific Advisor for ADME-Tox Services here at BioIVT, and today I will present ADME 101 CYP Inhibition. Let's start with a quick refresher of ADME, which stands for Absorption, Distribution, Metabolism, and Excretion. Understanding ADME characteristics of a drug candidate is important in drug development, as that helps to predict how a drug behaves in the body. And today, we will focus on metabolism and particularly on the effects of changes in activity of drug-metabolizing enzymes. Before we go into the details, let's understand where CYP or cytochrome P450 enzyme inhibition fits within the typical in vitro drug-drug interaction studies, or short called DDI, for small molecules. The main objective of drug development is to create drugs that are both safe and effective for patients, and that requires developing drug candidates that have desirable ADME properties. And these ADME properties are assessed in vitro DDI studies, which can be grouped into three categories as seen here: drug metabolism, drug metabolizing enzymes, and drug transporters. And today we will focus on drug metabolizing enzymes and inhibition. And in this context, typically two classes of enzymes are assessed: CYP enzymes and UDP glucuronosyl transferases, or UGTs. As the title of this video indicates, we will focus here on CYP enzyme inhibition. CYP inhibition studies evaluate the potential of a drug candidate to inhibit drug metabolizing enzymes, and specifically CYP enzymes. If a drug inhibits CYP enzymes, it can increase the risk for DDI. So let's take a moment to understand DDI in a little bit more detail and how that applies to CYP inhibition. A drug is called a precipitant if it affects other drugs that are administered at the same time. So, for instance, if a drug inhibits CYP3A4, it may slow the metabolism of a drug that has been taken simultaneously. As a result, the concentration of that drug in the body will stay high or higher for a longer period, and that elevates the risk of increased therapeutic action. In addition, there's also a higher risk for adverse effects, such as toxicity. So let's look at two well-known examples. The first one is ketoconazole, which is a potent antifungal and a strong inhibitor of CYP3A4. It can alter the levels of other drugs taken at the same time, leading to adverse effects. And indeed, three drugs, terfenadine, marketed as Seldane, cisapride, or Propulsid, and asdemizole, sold as Hismonol, have been withdrawn from the market because of ventricular arrhythmias when taken with ketoconazole. Another example is mibefadril, that was marketed as Posicor. It was a non-selective calcium channel blocker used to treat hypertension and angina pectoris. It was a long-lasting CYP3A4 inhibitor and pulled from the market less than a year after approval due to its causing toxic interactions with other drugs such as antihistamines, antibiotics, and statins. So now let's go back to CYP inhibition. Now, CYP inhibition studies are recommended by regulatory guidance such as the ICH M12 as part of the in vitro DDI package. Here at BioIVT, we routinely evaluate three types of inhibition: direct inhibition, time-dependent inhibition, and metabolism-dependent inhibition. And I will explain these types in more detail later. What I do want to do now is, though, point out that time and metabolism-dependent inhibition are not always distinguished, such as, for instance, in the ICH M12, where both are called time-dependent. Now, the results and conclusions from CYP inhibition studies are crucial. They determine the CYP inhibition potential, which is essential to get a better understanding of the clinical DDI potential. Additionally, this data will also assist with the design of the clinical DDI studies should they be needed. Let's now look at the typical design of CYP inhibition studies as illustrated in this table here, which closely reflects the setup here at BioIVT. There are two study designs dependent on the objectives. The first is a screening or compound ranking study, and the second type is a definitive or IND-enabling study that meets the requirement for regulatory guidance, regulatory submission. Now, starting from the top, the test system is typically pooled human liver microsomes for both study types. With that said, in certain cases, pooled suspended, or in very rare cases, plated hepatocytes may be used. 7 CYP enzymes are routinely evaluated. These are CYP1A2, 2B6, 2C8, 2C9, 2C19, 2D6, and 3A4 and 5. For the simpler and more cost-effective screening study, a cocktail substrate is used to assess all 7 CYP enzymes simultaneously. For IND-enabling studies, each of these 7 enzymes is evaluated using an individual substrate. Typically, 7 concentrations are used to calculate an IC50 if inhibition is observed. Now, with that said, in some cases, it might be sufficient to use just 1 low and 1 high concentration to demonstrate the absence of inhibition. Now, with regard to replicates, 2 are generally considered adequate for the standard setup using human liver microsomes. It is important to note that the protein concentration in the assay is critical and needs to be low, as higher protein concentrations can lead to artifacts. In addition, substrate incubation time is kept short to determine inhibition under initial rate conditions. Now, as mentioned earlier, 3 types of inhibition are assessed: direct, time-dependent, and metabolism-dependent. And positive controls are, of course, included: an inhibitor cocktail for screening studies and CYP enzyme-specific inhibitors for the IND-enabling design. Last, the detection method for the substrate is LC-MS-MS, and the deliverables include a report with IC50 value calculations if applicable. So now, as promised, let's now briefly explore the different types of inhibition and delve a little into some of the biochemistry. First, and shown here on the left, direct inhibition. This type of inhibition is reversible. So, for instance, the inhibitor binds to the same active site as the substrate. Now, direct inhibition is evaluated by adding the substrate, NADPH, and the drug candidate, which serves as the inhibitor, at the same time. If the CYP enzyme is inhibited, the inhibition constant Ki is determined. Now, typically, if the substrate concentration in the assay is close to the Km of the CYP enzyme, the IC50 can be divided by 2 to calculate the Ki. Now, this method was indeed developed by my colleagues here at Sinotech, now part of BioIVT, with Lois Haupt as the first author of the publication, which was cited in the FDA guidelines. Now, moving on, the other 2 types of inhibition are often collectively referred to as time-dependent, but they are mechanistically different, and I strongly believe it is worth distinguishing between time-dependent and metabolism-dependent inhibition, at least in liver microsomes. Time-dependent inhibition is evaluated by pre-incubating the drug candidate with the liver microsomes for 30 minutes without NADPH, followed by the IC50 determination. If there is a decrease of the IC50, this indicates non-enzymatic degradation or metabolism by non-NADPH dependent enzymes. So further evaluating these possibilities might be a possibility. Lastly, there is metabolism-dependent inhibition. This is the mechanism when an IC50 decrease is observed after a 30-minute pre-incubation in the presence of NADPH. With few exceptions, this happens due to 3 different mechanisms which can be distinguished running BioIVT's so-called reversibility assay. First, reversible metabolism-dependent inhibition. This means that a metabolite is a more potent direct inhibitor than the parent drug candidate. Second, irreversible metabolite-dependent inhibition, which means that a metabolite binds irreversibly, or in other words, covalently to the enzyme. An example for this type of inhibition includes the strong CYP3A4 inhibitor mibafetril, which I mentioned before, and which was removed from the market for that exact reason. And third, quasi-irreversible inhibition, which refers to the event when a metabolite binds tightly to the heme of the cytochrome P450, forming a so-called metabolite inhibitory complex. Examples for this type of inhibition are erythromycin, clarithromycin, and S-fluoxetine. In this context, it is important to emphasize that irreversible inhibition implies that the inhibition will persist even after the drug is removed from the body and until new enzyme is synthesized, which can pose challenges for further development. Now, if irreversible or quasi-irreversible metabolism-dependent inhibition is observed, we normally recommend following up and determine the unbound inhibitor concentration causing half maximal inactivation, called K∆I, and the maximal inactivation rate, K-inact. So let me briefly go into the assays associated with metabolism-dependent inhibition. First, the reversibility assay. While not required by regulatory agencies, this assay is scientifically valuable for further drug development as it will provide insights into the inhibition mechanism. And here's a very simple scheme that describes the principle of the assay. The drug candidate is first pre-incubated in a buffer in the presence of NADPH. Now, one sample will be treated with potassium ferricyanide to distinguish between quasi-irreversible and irreversible binding. If a metabolite inhibitory complex has formed, the ferricyanide will disrupt the bond to the heme moiety of the CYP enzymes, and the enzyme will regain activity after ultracentrifugation to separate the free protein. In a separate incubation, the sample will be subjected to ultracentrifugation to remove unbound drug. If the inhibition is reversible, the protein will be active, whereas in the case of irreversible inhibition, the protein remains inhibited. Now, if further evaluation of metabolism-dependent inhibition is needed, Ki, so the unbound inhibitor concentration causing half maximal inactivation, and K-inact, the maximal inactivation rate, need to be determined. This study is conducted again in pooled human liver microsomes using 6 concentrations of the drug candidate with a slightly different setup than the standard design. A key difference here is the varying length of the pre-incubation time points, with typically 5 time points being assessed. Additionally, this setup uses a higher protein concentration, so it might be beneficial to determine microsomal protein binding. Now here is a reference table of CYP enzyme-specific substrates and marker reactions that are routinely used and recommended by the ICH M12. The first column lists the 7 routinely assessed CYP enzymes. In the next column, we have the substrate or marker reactions. And then the last 2 columns list the positive controls for each of the 7 CYP enzymes, with one for direct inhibition and another for time-dependent or metabolism-dependent inhibition. With that, let's look at the deliverables of CYP inhibition studies. Here's an example of the curves that are generated. These two graphs show the CYP inhibition curve for CYP3A4/5 generated in human liver microsomes with testosterone as a substrate. In both graphs, the x-axis represents the concentration of the drug candidate being tested, while the y-axis is the rate expressed as the percentage of the control. If inhibition occurs with increasing concentration, the rate will decrease. Now on the left graph, the blue line represents 0 minute pre-incubation, so showing the results for direct inhibition. And in red is the 30-minute pre-incubation in the absence of NADPH, so time-dependent inhibition. As you can see, there's barely any change, and both curves show modest inhibition with an IC50 of around 7 and 10 micromolar, respectively. On the right graph, the same red line from the left graph serves as a comparator for the green curve, which represents the 30-minute pre-incubation in the presence of NADPH. The shift to the left is evident, and the IC50 is indeed lower and was calculated to be around 2.8 micromolar. And here are two examples of drugs that are CYP inhibitors. The first example is ritonavir, marketed under the brand name Norvir. This is an antiretroviral used for HIV infection treatment and is a strong direct CYP3A4 inhibitor. It is indeed used on purpose to reduce the CYP3A4 activity to increase the levels of other HIV medications. The second example is, again, mibeverdril. I've already mentioned this before, and indeed, mibeverdril is a strong 3A4 irreversible inhibitor, which was withdrawn from the market due to its adverse effects and potential for fatal DDIs. So finally, let's discuss the interpretation of CYP inhibition results. The overall objective is to determine whether clinical studies, DDI studies, are needed, and there are equations in the regulatory guidelines to do so. First, the risk for reversible CYP inhibition can be excluded based on in vitro data if the unbound maximal concentration Cmax at the highest recommended dose at steady state divided by the unbound Ki is greater than 0.02, or differently said, if the unbound inhibition constant is greater than 50 times the unbound Cmax. For orally administered drugs, the intestinal CYP3A inhibition needs to also be addressed, and the risk can be excluded if the unbound Ki is greater than 0.1 times the maximal dose divided by 250 milliliters. Lastly, the risk for irreversible inhibition can be determined using the following equation. The observed apparent first-order inactivation rate, called Kops, plus the apparent first-order degradation rate constant, divided by the degradation rate constant. If that value is less than 1.25, the risk can be excluded. And Kops, in turn, can be calculated as shown here using the equation on the right. With that, let me summarize with 4 key takeaways from the CYP inhibition studies. CYP inhibition studies evaluate the potential of drug candidates to inhibit CYP enzymes. Now here at BioIVT, the study design is set up to evaluate the potential for direct inhibition, time-dependent, and metabolism-dependent inhibition. And typically, 7 CYP enzymes are evaluated that are recommended by the regulatory guidance. Last, results and conclusions of CYP inhibition studies will aid in understanding the DDI potential and assist with the design of clinical DDI studies. With that, we are at the end of the short introduction into CYP inhibition studies, and here are some additional resources that can be found on the BioIVT website. Thank you so much for watching, and please reach out if you have any questions or any needs for CYP inhibition or other DDI studies. Thank you.